I think we're going to get started. I'm Max Leisterson. I'm a postdoc here. And our next speaker is actually the first time I've introduced him. He's introduced me a few times. It's Ben Raphael from Princeton. He's going to tell us about cancer genome evolution. Ben?
There we go. Thank you, Max, for the introduction, the invitation to speak. If you were coming to hear Bill Hahn, you're probably going to be disappointed the next half hour because there's not going to be any large-scale CRISPR screens or RNAi or really cool data sets of the type that Bill and his group generate. Instead, you're stuck with a little bit of algorithms and with me. But I'm now the only cancer talk, so that's sort of a plus for Bill, unfortunately not being able to make it today. And that means that I need to give you a little bit of introduction. And of course, cancer genomics is a huge topic. But for the purposes of this talk, we're really going to focus on the DNA, on the genome. And as I'm sure most of you know, cancer is driven largely by somatic mutations that accumulate in the genome over the course of multiple cell divisions. And so sort of classic clonal theory of cancer is that from a single founder cell, which has accumulated mutations, and somatic mutations essentially accumulate every time cells divide. Most of them are benign passenger mutations, but a cell that acquires some driver mutation, a functional mutation that might give this cell a selective advantage compared to other cells in the tissue will then start to grow faster than its neighbors, start to overtake the tissue, and then subsequent rounds of mutation and additional driver mutations give the tumor the various terrible, nasty properties that tumors have, being able to invade and metastasize and escape the immune system, et cetera. So this picture has largely been known for decades, and there's a review article from Peter Knoll that has a nice picture like this from the mid-'70s. And what's happened, as most of you know, in the past few years, is that through the application of large-scale genome sequencing, we can start to quantify this picture. And so immediately some questions come to mind. Which mutations are present in a tumor sample when you go and sequence it? Which of those mutations are drivers and which are passengers? Do all the cells in the tumor have the same driver mutations? Actually, we sort of know that any tumor sample will have mixtures of normal cells and the tumor cells within them can have different complements of somatic mutations. Does that heterogeneity matter in terms of the tumor's ability to grow or be resistant to drugs, et cetera? And then we could start to ask sort of evolutionary questions. In what order did these mutations arise? The sort of evolutionary process, can we quantify it? And so we, in my group, are computational, and we develop algorithms to try to address these questions. And we work in sort of all of these different areas. And there were essentially two talks that I had thought about giving. One was on our work in answering the second question by looking at combinations of mutations in pathways and networks. And I realized that Max Leisterson, who's here, was formerly in my group and has done a lot of the work in recent years on that topic. And so I thought folks at Microsoft have heard Max present, and he's presenting again apparently next week. Those of you that haven't heard this work will have to hear it from Max or from me another time. And instead, I decided to focus on our work on these second two points. So thus, the theme of cancer evolution. So I should say that I'm going to focus on the computational and the algorithmic problems here, but ultimately the goal in answering these questions is to understand cancer biologically, but ultimately in answering these questions, we hope to be able to improve diagnosis and therapy of cancer. And I'm not going to talk about how that link works in today's talk. So what's important about understanding cancer as an evolutionary process? Of course, there's a basic science reason. It's sort of nice to be able to piece together what happened in the past, as evolutionary biologists do. Can we piece together what happened in the past when we observe a present-day tumor? And that's sort of interesting for understanding the dynamics of this process. But by understanding this process, it also can help us therapeutically. And one of the sort of interesting examples of that from a few years ago, and there are many others, but this perhaps was one of the first. In AML, the group at WashU Genome Sequencing Center sequenced a primary AML sample and then a relapse. And then what they went and did was they sort of computationally separated out this primary tumor sample into multiple subpopulations of cells that had different complements of somatic mutations. And what they observed in the relapse was that following treatment, it was a clone, a subpopulation of cells that was really present in only a small percentage of the tumor sample, which was the one that grew out and formed the relapse. So understanding this structure can be important for how we diagnose or how we treat, as we get more targeted agents, cancer patients. So there is sort of a clinical angle to this as well. Okay, so cancer is an evolutionary process. There's a computational biology audience, so of course you know evolutionary process we describe via a tree, phylogenetic tree, right? So, turning this picture on the side, here's our founder cell, and it's grown out and formed these additional mutations. The colors are not so great here. Maybe they're better on this version. And we can describe this process of cells dividing and acquiring mutations via this mathematical representation of this tree. Here the tree could be individual cells, if we have that resolution, or it could be subpopulations of cells that share the same somatic mutation, so-called clones, if we have sort of less resolution to resolve single cells. So the idea then, if we sequence our tumor at the present time, we measure these cells are clones, can we go back in time and reconstruct the tree? And this is classic phylogenetic tree reconstruction. I'm sure many of you have studied it. The one-slide version of this is to think of describing a genome, in this case a tumor cell or a clone, as a sequence of characters. These characters could be, in our case, they're going to be somatic mutations. More generally, if these were species, they could be even some qualitative traits that we have. And so we measure one of k possible states for each character. So our input data is for each character its state. So here, if I have characters with 2 states, 0 and 1, then for each species or taxa, I have its state for each character. So this character state matrix becomes the input. And what we do then is we try to find a tree whose leaves are labeled by the rows of this matrix, the individual species, whose internal nodes are labeled by some ancestral states, such that the resulting tree minimizes some objective function on the evolution. Okay, and so the classic ones are maximum parsimony. We want the tree such that we can describe our input data on the leaves by the fewest number of changes in state characters. Or if we have a probabilistic model for our evolutionary process, we can compute maximum likelihood, compute the total probability of all the changes we observe on our tree and try to find the one that has maximum probability. Classic phylogenetics, right? This is stuff that I'm sure most of you know and have learned before and probably run many of the fancy algorithms to do this. So, why are we talking about this in the cancer context? We can just plug this in. This has all been well developed. Well, we immediately are faced with a problem when we apply this to most cancer data that's been generated, which is that most of the data, nearly all of the data, is from bulk tumor samples. Meaning we haven't sequenced single cells, we've sequenced a tumor sample, tens of thousands, millions of cells, so it's a mixture. So instead of measuring the individual leaves of the tree, computationally what we've measured is a mixture of the leaves of the tree. So these algorithms don't apply to mixtures, they apply to the individual leaves. So could we, how can we address this issue? So this is one of the first challenges in sort of cancer phylogeny, and that's the first one I want to describe. And then the second one is that cancer genomes are very complex. They have somatic mutations across all different genomic scales. So on the small scale, you can have single nucleotide variants or single nucleotide changes in single letter of DNA. You can have but you can have much larger changes. Again, it's sort of washed out here, so you don't see the pretty colored pictures from this chromosome painting or spectral karyotyping, where you see individual chromosomes with genome rearrangements, the sort of older cytogenetic technique. Now we sort of build pictures like this computationally from genome sequencing data. So this is what's called a Circos plot. This is a breast cancer sample that was sequenced a few years ago, and these are the 23 chromosomes arranged around the circle. And what they're sort of showing here are these green edges are these translocations that you would observe here. So you can see these extensive rearrangements. These blue ones are also sort of links on the same chromosome, large-scale rearrangements. But then you have these sort of copy number changes, which is shown on this axis where we're measuring the density of reads that map to a particular region. You can see amplifications or deletions as you go along here. So changes on all scales, from the single nucleotide all the way through large-scale gross chromosomal rearrangements. So in phylogenetic terms, what should we use as our characters? Which mutations do we want to analyze? And that choice actually matters in terms of how we might be able to reconstruct the genome. So let's start with the simplest model. Let's assume for now that we're only going to look at single nucleotide variants. Yeah, okay. And so for a single position in the genome, we'll describe it via 1 if it's mutated and a 0 if it's not. And let's assume that we've sequenced individual cells in our tumor sample, which now is becoming possible, and there are great data sets that have done this, but it's a small minority of the cancer sequencing that's done. So by doing so, then we get this great character state matrix. So here we have, again, each of the leaves is one of our cells. We've measured the individual characters, here the individual mutations, presence or absence of each mutation, and now we can apply choose your favorite phylogenetic algorithm, maximum parsimony, maximum likelihood, and build the tree. Let's do the simplest one possible. The simplest possible phylogenetic model is what's called the perfect phylogeny, and here it's 2-state because each of our characters has 2 states. And this model makes a very a somewhat strong assumption on the evolutionary process, and the so-called infinite sites assumption, which is that we assume that a mutation occurs at most once at a given position. So if we think of the somatic mutation process, single nucleotide mutations are what we're analyzing. So what we're saying is, over the course of the tumor growing and mutating and dividing, at an individual nucleotide on the genome, a somatic mutation happens at that position only once. Okay? Now, this turns out not to be such a bad assumption for the cancer process. And again, it's sort of hidden in the name here, infinite sites, because the genome is long, 3 billion nucleotides. There aren't maybe so many cell divisions. So by random chance, the probability that we would have a mutation at the exact same position is really low. So we're going to say it's 0. That's how we get this assumption. So the nice thing about, if we make this assumption, the perfect phylogeny, is that we can compute this tree in essentially linear time in the size of the input. So this is the simplest possible phylogeny problem. We could find the answer really fast. In general, maximum parsimony, maximum likelihood are harder problems. In fact, what are called NP-complete problems. But here, the simplest thing possible. Okay, so that's great. Here's the simplest model. If we measured single cells, we could do it very efficiently. But what happens if we mix? So now if we mix, instead of measuring at an individual position whether or not a mutation is present or not, instead what we really end up measuring is, when we look at the reads that cover that position, we instead get a measurement of which fraction of those reads contain the mutation. So now our 0-1 measurement at a location of the genome, mutation present or not, has become this analog measurement, which is which fraction of the reads tell me that their mutation is present. This is the so-called variant allele frequency. And this variant allele frequency is proportional to the fraction of cells that have the mutation. Of course, hidden in this fraction is some tricky stuff, and for those of you that have done this before, you'll know that it depends on the number of copies of that region. So if you have these copy number aberrations, it'll change this proportionality constant. Let's forget about those complexities to start. Okay, so now we've got this analog measurement. We've mixed our data. So in terms of the tree, now instead of this 0-1 character state matrix, we get these values between 0 and 1, each of which represents somehow a mixture of the leaves of this tree that I'm not observing. Okay, so trying to sort of understand this process, a couple of years ago, I postdoc Mohammed and working with a former PhD student now, Allele, said, could we reconstruct this tree from mixtures of the leaves? Okay, so what we've done here is this data, this F, is generated by some tree which we don't know. What I've done here is taken multiple samples from my tumor, and each of those samples is some mixture of cells from this tree in certain proportions, which we also don't know. So here, sample 3 is 80% this cell with these 3 different mutations. Unfortunately, the colors are washed out here. And there's also some normal cells, which are indicated here by no mutation. So 80% this and 20% normal, et cetera, et cetera. Sample 1 you can see is a mixture of 3 different cell types. Okay? So could we directly from F get this whole picture, the tree plus these proportions? Seems actually quite hard to do. We're missing a lot of information, but we can actually write this down in a very simple way, forgetting about any uncertainty in the data. So this is important. Let's just be good computer scientists rather than statisticians. Forget about uncertainty in the data. Let's just look at the underlying structure of the problem. So we could describe these frequencies, the observed frequencies, we can describe them in a very simple way. They're supposed to be mixtures of the leaves of the tree. If I make this perfect phylogeny assumption, there's actually a one-to-one correspondence between trees and 0, 1 value matrices, where each entry here just indicates for each leaf which mutations are present or absent. There's a nice way to transform a tree to these 0, 1 value matrices. And then all we're going to do is each sample is just mixing the rows of this matrix, and the proportions in which they're mixed are given by the rows of this mixing matrix. So if I multiply this matrix B times U, I get F. Okay? What we want to do is the reverse. Given F, can I factorize it into B times U? Right? So this is like you learned in elementary school, given a number, can you factor it into primes? Except here it's a very structured problem involving matrices and special classes of matrices. Okay? So the sort of formulation, which we wrote down in a paper last year, is sort of floating around in a lot of other papers, and this idea of phylogenetic mixtures has become very popular in the computational community, and so you can find sort of different versions of this problem written in different ways in a bunch of different papers, but what we were able to do is actually completely characterize the solutions of this problem. Okay? So the way we do that is by looking at these frequencies, and it turns out that these frequencies actually encode information about the tree in a very special way. So the first condition that one can think about is if a mutation is ancestral to another mutation. So here this gray mutation happened before this orange mutation. Now, mutations happen only once on the tree. So what that means is every cell that has the orange mutation has to have the gray mutation. The gray one's ancestral. Every cell with the orange mutation has to have the gray mutation because gray is ancestral. So what that means is the frequencies of the gray mutations have to be bigger than the frequencies of the orange. There's actually a condition now which is telling us somehow links between the observed frequencies and these conditions. A sort of slightly more involved condition, they won't really derive, is actually sort of taking this idea and generalizing a little bit more, which is to say that the frequency of a parent has to be greater than the sum of the frequencies of the children. Okay? So these are two conditions that are somehow linking the observed mutations to the unknown structure of the tree. And what we can do is take these conditions and we can build a combinatorial object, a graph, directly from these conditions, and we can write down all the solutions as certain structures in this graph. So in brief, what we do is we can build a graph that has an edge between two mutations if one is ancestral to the other. These directed edges are just derived directly from this frequency condition. This is a directed graph. This graph encodes within it all the possible trees that are consistent with the frequency matrix and encodes them in a very special way. All we need to do are find what are called spanning trees, so subsets of edges that form a tree, and they just have to be special spanning trees that satisfy the sum condition. So just from the frequency matrix, just by looking at one condition here, a very simple condition, I can write down this graph. That graph encodes all the trees that are consistent with the frequencies. It's a very nice, beautiful framework, and you can show that that's actually all there is to this mixing phylogeny problem. Now, there's actually something kind of also interesting that happened. This perfect phylogeny, the simplest possible phylogeny problem, which we could solve in linear time when we measured the leaves, when you mix them becomes an NP-complete problem, a very hard computational problem. So already, you can see how this loss of information in mixing makes phylogenetic problems much harder to solve. So, of course, when we look at real data, we have to deal with errors. And without going into all the details, what we can sort of do is relax these point estimates of the frequencies into posterior probabilities of the frequencies by using some model for the uncertainty in the measurement according to the number of reads that cover it. And so then we relax this condition about the ancestry of one mutation being ancestral to the other by this inequality in the frequencies into a statement about the probability distributions and how much they overlap. And we build a graph based on this uncertainty. And without going into all the details, we then find this spanning tree and this approximate ancestry graph. Okay, so just to show you one sort of example of applying this, we took data from a renal cell carcinoma sample that was chopped up into about a dozen regions. And then we built this tree this mixed tree from the data. So what you can see, here's the tree in black. These are describing groups of mutations. And in red here, these are the regions. So each region that we sequence is some mixture of the leaves of the tree in the proportions indicated on these arrows. Now, this sort of shows you the difference between the type of analysis we're doing here versus the type of analysis that was published in the paper, where what they did was they took each region, assumed that it was single clone, and just plugged it into standard phylogeny approaches. So each leaf in their tree is a single region. They're assuming that that region is all the same. Here you can actually separate out the mixture. So for the aficionados and people who work in this area, I'm not sure if there are any, but if there are, then you might have seen other ways to do this where you really just look at clustering the mutations according to the frequencies in different samples, and you do this using Dirichlet processes or choose your favorite clustering algorithm. In essence, what we're doing here is using the tree to find an extra constraint on how these clusters should be arranged. The clusters shouldn't be arranged randomly. They're actually correlated by the structure of the tree. So that was for 2 states, which is nice for perhaps analyzing single nucleotide mutations. When you go to beyond 2 states and you want to look at copy number aberrations, which are very common in tumors. So here's an example of a copy number plot where in the different chromosomes we've looked at how many copies there are across the genome. Now you need to do something more complicated. You can now have many possible states. Here's your 2 a single locus. You have 2 copies of each chromosome. It could be non-mutated. You could have a single nucleotide variant on one copy but not the other. You could amplify one of the chromosomes, make a second copy of it. You could have that with or without mutation. So you now have multiple possible states for each locus. The whole thing can become very complicated, and you need now an evolutionary model to deal with more than 2 states. And the simplest such model you can do, the sort of generalization of infinite sites, is to do what's called the infinite alleles model, which is to say that a character can change more than once, but it changes to a state at most once. So in the DNA case, for example, you can change from A to C or A to T or A to G, but you'll never go back to the same state. So it slightly generalizes that infinite sites model. So turns out that the theory sort of carries over. It gets a little more complicated. Instead of having this matrix with zeros and ones, now you've got multiple states for each possible thing. Yeah.
Assumption about the to a state.
Yeah.
Because you could have an amplification over a large region,
Yeah.
which then also undergoes a deletion over a partial set of that region.
Yeah.
And then you would return to copy number neutral in that overlapping region. That doesn't seem to have such a low probability of happening, but that would actually contradict that assumption.
Right.
Could you repeat the question on the mic?
Yeah, sorry. So the question was, well, once you start allowing copy number aberrations, they could span multiple long regions of the genome that contain different single nucleotide variants, and they could overlap in complicated ways. So I could amplify a part of the genome and then delete a subpart of that genome and affect this single nucleotide variant in all sorts of unpredictable ways. Yeah, so the answer is yes, and you're pointing to the other big complication in dealing with cancer phylogeny, which is that the set of operations is actually very large. And so these copy number aberrations are very messy. And this was the point I wanted to touch on quickly in the last part of the talk, assuming I'm talking pretty slowly, actually. So that'll be very fast. But yes, so this is sort of a slight simplification in handling copy number aberrations. So we've generalized it a little bit. We allow multiple states. But we haven't fully done everything yet. But the theory carries over somewhat nicely in that now if you have multiple states that you could have per character, so here are zeros, ones, and twos, then this frequency matrix then just becomes this, sort of you measure a frequency for each mutation in each sample in each state. So now you've got this sort of slices of frequencies, this sort of frequency tensor, if you will, and you then have to analyze this structure, and it turns out that you can sort of take the same formulation almost, this matrix factorization problem, now with this more than 2 states. What you end up sort of describing that is as multiple binary matrices that are all mixed together with the same mixing matrix U. So in this factorization sense, what you get is that given this matrix, this now tensor F, you want to factorize it using the same U. So it's sort of a simultaneous matrix factorization problem. And the rest of the theory sort of carries through, and it gets somewhat complicated, and I'm not going to sort of go through all of it, just to say that then you can make this sort of practical and run it on data by taking uncertainties in these variant alleles, taking the copy numbers that you would estimate, and you need these proportions, and you can do this in various ways. I won't describe. And then for each of those data, you can actually generate multiple possible frequency matrices, tensors. This is actually getting towards some of the ambiguity that you're pointing out when you have these copy number aberrations, that there's ambiguity in what the frequency of the individual mutation should be. It depends on if you interpret it as deletions or amplifications or overlapping in different ways. So some of that's encoded here. And then each of those, you have even more ambiguity, and you can get lots of different trees that are consistent with the data. So this algorithm, which we call SPRUCE, which is an acronym for the sort of combinatorial on-enumeration, allows us to look at many, many different possibilities. And what one finds is that there tend to be, once you allow copy number aberrations, not surprisingly, many, many ways you can reconstruct the data from this mixed frequency matrix that are all consistent with the data. So pointing at one tree is not necessarily the best thing to do. And just to show sort of one version of that, this is a very complicated data set that was published earlier this year of a metastatic breast cancer, and they're sort of showing multiple samples throughout the body that were sequenced, and they tried to sort of reconstruct the complicated relationships between them. And they are, in fact, very complicated. But when you look at the sort of tree that is published there, what you can sort of see is that, in fact, there are many, many alternative trees that are consistent with the data, and those trees start to sort of shift around the ancestral relationship between different nodes. So now if you go back to this idea of, let's treat a tumor by looking at the different populations of cells that we have there within it, when you start changing the composition and changing these ancestral relationships between nodes, it might change how you would think about the tumor and how you would decide which subpopulations you should target with drugs. So I think I'm out of time, pretty much, right? Close to it. The lunch is waiting, though, isn't it? Oh, okay, good. All right. So I'm just going to really quickly say something about this last piece. So this was the summary of the part in terms of mixing trees. And the sort of take-home message here is that by looking at cancer data, you get this sort of new class of phylogenetic problems, which we hadn't seen people really address before, which are problems of take a phylogenetic tree and mix the leaves, and can you reconstruct it? We did it with the simplest model, perfect phylogeny. We did it with the next simplest model, multi-state perfect phylogeny. But now you could ask the question about, what about maximum parsimony? Or what about various likelihood models? When you mix them, can you still recover anything? And under what conditions can you mix them? So it's sort of a fun computational area, but at the same time has important implications for how one might look at cancer data. So in a couple of minutes, and I actually, for the sort of more mathematically inclined, I mean, everything I sort of described in these first two parts was really just special classes of matrix factorization problems. We're given this frequency matrix, written as U times B. Depending on the structure I put on B, I get different problems which I can solve or potentially not solve, and some are easy and some are hard. So it's also sort of an interesting theoretical area as well. Okay, so quickly just to say something about this issue with complexity of cancer genomes and with copy number aberrations. The question here pointed out actually one of the other really challenging part about analyzing cancer genomes with phylogenetic methods, which is that if you look at various classes of mutations that can happen, and in particular if you look at copy number aberrations. So here I'm thinking of the genome now as segments, where each segment has a different number of copies. I can infer those copies by mapping reads to the genome and then seeing the density of reads that map and try to infer these integer copies. But the point is these changes, these duplications and deletions of regions of the genome happen frequently. So could we actually incorporate the copy number, and this shouldn't say the copy number as phylogenetic characters? Okay? Can we actually analyze those as events as opposed to just mutations as events? And this is exactly the question that was sort of raised, which is that almost all of this work in phylogeny really makes the assumption that characters evolve independently. And so you can analyze them independently. But when you start looking at copy number aberrations, so if I think abstractly of a cancer genome as these segments with different numbers of copies, then copy number aberrations, like here a deletion or various duplications, affect multiple characters simultaneously. So they can't be analyzed independently, and you need much more sophisticated methods even to just compute, say, the minimum number of such operations that transform one string into another. And so this is sort of a simple model that was published a couple of years ago to analyze cancer genomes, where we have a very simple version of what copy number aberrations do. They either reduce the number of copies of a genome, of segment of the genome by 1, or they increase them by 1. And so you try to compute the minimum number of such operations to transform one string into the other. Turns out you can do it in linear time via a paper that was published by Ron Shamir's group just a few months ago. So when you look at a model like this, when you try to actually analyze these more complicated events, note it's not reversible, and this idea that the characters are not independent because a single event affects multiple characters simultaneously. Something that doesn't happen with single nucleotide mutations, right? If you look at a single nucleotide mutation, it's like, even if it has back mutation, it's only affecting that one locus. Here, the event affects multiple characters. So can you go further? And these are sort of things we've been interested in and working on. So a PhD student, Simone, was recently sort of trying to extend this idea to build trees from these copy number profiles. So the idea here is that you would build a tree, you've measured the leaves, you then try to infer the tree that minimizes the number of these segmental duplications and deletions that occur on the tree. This is hard problem. We speculate it's, again, NP hard problem, but we had an ILP version an ILP solution to solve this. Could you mix a problem like this? So now if you measured individual cells and their copy number profiles, do this. Already this is hard. Could you mix it? And mixing it actually gets really complicated, very complicated. And instead of going through all of that in the interest of time, and since lunch is happening, let me just sort of show you the end result here. So Simone, working again with Mohammed, sort of put together a version of this where various things going on here. You've got multiple samples of your tumor. You've got these copy number fractions. They're non-integral because they're mixed, right? Because each sample could include multiple cells with different, copy number profiles. So now you have these mixed profiles. And then what you'd like to do is infer the tree that describe those profiles and how they're mixed in each sample in order to describe the data. So given just the copy number fractions, could you get everything? These profiles C, which are now going to be these integer profiles, and how they're mixed, as well as the tree that describes them. Not surprisingly, it turns out to be a hard problem, we think, but we were able to sort of work out an optimization framework that allows us to try to, for given frequencies f, try to sort of minimize the error subject to some cost on the tree, and it's just a sort of coordinate descent type algorithm where we optimize u, and that's just a linear program. It can be solved easily. Then we optimize c for a fixed u, and that's an ILP and can be solved easily, and then you bounce back and forth between these steps. So just to give you a sense of the results here, and this is sort of the last slide for those of you that are hungry. There's just a comparison of how this works and how using the tree can really get you extra information. So just look at the three parts here. This IMF, this is just if you just try to do factorization without any tree. And so this vertical axis here is the error that you have. And so you can see that the errors are very high for not using the tree. But the errors decrease as you get more samples from your tumor. So if I chop up my tumor into more mixed samples, there should be more information. And then you can sort of see that I can get the tree better in blue. And then if I instead have the tree but don't deal with the mixture, that's this color, which I can't even tell what that is. It's purple. You wouldn't know that in this mixed slide, but it's purple. So that's here. And you can see that the purple actually, as you sort of increase the number of samples, doesn't really gain much information. And that's because it's sort of not using the fact that those samples are mixtures. It's treating them as if they're all the same. But then if you do everything together, and that's this red one that, again, you can't quite see. For 2 samples, you're still doing really well, much better than if you didn't use the tree. For 5 already, you're getting it a lot of the time. This is over 20-some simulations. By 10 samples, pretty much every example, you're getting the tree exactly. So this sort of shows by using these 2 constraints by using the structure of the tree. You get a lot more information, and then you can apply this to real data. And this is this same prostate cancer paper where they published some trees, and the trees are very complicated that they inferred mostly from single nucleotide variants, and then put some copy number aberrations on the root of the tree. We get many of, on the first branch of the tree. We get many of those same copy number aberrations in the first root, but we see many, many subsequent copy number aberrations that split up the tree into these different clones, which are then mixed together. So that's the end, and I, again, just wanted to emphasize the fact that there's a lot of interesting problems here computationally. So this mixture problem, but then dealing with the various difficult types of mutations that occur in tumors really has to address this problem. The characters, particularly for copy number aberrations, and I haven't said anything about genome rearrangements, are really non-independent. So a lot of the standard phylogenetic algorithms you can't just apply directly. And so there's a lot of work to do here, both computationally and in terms of turning some of these more theoretical models into practical algorithms that you can use to analyze data and to say things about the composition and evolution of tumors. And so I'll end there and just acknowledge the folks in my group that did the work here in bold, groups that, for the other part I didn't talk about, and the groups sort of slowly migrating from Brown to Princeton. So we're kind of in two places at the moment. And software for a lot of this stuff is available. So if you actually are analyzing cancer data, I encourage you to go take a look there. And thank you for your attention.
Thanks, Ben. We have time for a couple questions.
Hi, I was just wondering, you get ensembles of trees as an output, is that right? So
yeah.
Would that be possible then to see whether there are commonalities on the ensembles and see what say, ancestors of, say, high confidence, they occur a lot, what paths occur a lot, and that sort of thing as well?
Yeah, it's a great question, and that's, I skipped over that part, but that's exactly what we do to try to summarize the output of the algorithm where there's lots of ambiguity. So, when you have copy number aberrations, lots of ambiguity, lots of trees, but many of them tend to have common features. And certainly the events that are early in the tree that are shared by essentially all cells in the tumor, so-called clonal events, seem to be pretty well conserved across trees, and then things get messier as you go down towards the leaves. So yeah, it's a great suggestion. We do some of
that. For your first example of the VAF factorization, you use this idea that we should expect the frequency of ancestors to be greater than the frequency of the daughter cells.
Yeah.
Can you just explain that more? I guess I'm confused in that, both these populations might be in a mixture and under selection, and you can imagine environments where maybe the daughter might have a selective advantage and be greater too.
Yeah, no, it's a great question, and I said it a little bit glibly because it's not completely obvious that property should hold because you mix things, right? And you might mix them in different proportions in different samples. But even if the daughter cell is selected and has a real growth advantage, and so it starts to increase in frequency, that daughter cell had the ancestor mutation in it, right? So as it increases its frequency, it's increasing the frequency of all the ancestral mutations that it contains. And that's where this perfect phylogeny or infinite sites assumption really helps you. Because if I throw away that assumption, then it's possible that the daughter cell mutates that same position back, right, to the ancestor state, and then increases in frequency. And now all bets are off, and lots of different things can happen. So that assumption is sort of key to making this mixture problem solvable.
Hi.
Sequencing methods to actually either validate or enrich this, because then you wouldn't get a mixture, right? I mean, you just get a point.
Yeah. So the question was, if we've thought about using single cell to validate or just to go directly with single cell. Yes, we've started to look at some of the single cell data that's out there, and it does help to validate some of the assumptions that we have here. The particular data sets I showed don't have matched single cells, so we can't validate those. And then we've also started to look at the problems of reconstructing trees from single cell data. The challenges there are slightly different. In principle, it's easy because you don't have the mixtures anymore. It's a completely different problem. The problem you have to solve there is just dealing with all the errors of single cell data and allele dropout and all of these sorts of things. And those tend to be more statistical issues and getting the error model right. However, the second part of the talk where I described this non-independence between characters due to copy number aberrations is where there are actually very interesting things to do with single cell data, and we're working on that aspect of it. So the next talk will include that.
Hi, I'm interested in the role of space as a constraint on the possible mixtures, right?
Yeah.
So and you're nodding, so I guess you understand what I'm saying. Like in the metastases example, for instance, right? Those are not homogeneous, but they probably have founding clones.
Right.
And maybe even in the in the case of like core biopsies, there's some constraint that's like softer that you could impose to make the problem easier.
Yeah, no, that's a great question. So to paraphrase, it was like, well, what about space? I mean, when you take different cells samples from a tumor, you'd expect some spatial correlation where the mixture, the clones that are present in the left part of the tumor are more similar than those present in the right part of the tumor. We didn't use that constraint here, and we've sort of gone back and anecdotally tried to sort of look at, did we capture some spatial constraints? The data often isn't so great for where things came from within the primary tumor. Technologies that sort of sequence in situ will be really interesting for that. Maybe George Church will talk about that stuff this afternoon, I don't know. But we do get we do have some correlations like this. So for example, I won't flip back to the slide, but in this renal cell carcinoma that we analyzed, a couple of those samples were actually lymph nodes. And what we saw was that the mixing and the clones we saw in the two lymph node samples were more similar than those from the primary tumor. So anecdotally, it seems like yes, and if you had better spatial resolution, it's definitely an interesting and important constraint.
Let's do one more question.
You can imagine two different mutations that individually are not drivers, but one is only a driver in the presence like of the other one in some sense. So is that easily captured in these phylogeny trees? What I'm really asking is, does one node, going from one node to the daughter, can that sort of take into account multiple mutation events, or does it have to be just like a single character difference?
Yeah. So most of the time, this variant allele frequency that you use to look at a single mutation, the number of reads that cover a position, is a very noisy measurement, as you could imagine, of the fraction of cells that have that mutation. So you end up doing is actually grouping mutations together if they have similar variant allele frequency measurements across samples. And what this does is two things. I mean, it sort of increases your power to get a better estimate of that frequency, but it also reduces your resolution, if you will, to not being able to infer any relationships between mutations that you group together. So it's sort of a fine art as to how much one should do this clustering before trying to build the phylogenies. If I over-cluster, then I'm going to lose ancestral relationships. If I sort of under-cluster ahead of time, then I might start getting noise in the tree. To some extent, with mixed data, there are just going to be limitations in what you can infer, and that's where single cell would perhaps give you more information.
All right, so I think now let's break for lunch. We'll reconvene here at 1:30, and there are posters along the outside here, which you can visit during lunch or after. We'll have some time for a poster session. Let's thank Ben again.